Understanding the architecture of DNA originally required researchers to build physical, three-dimensional models to visualize the shape of the genome. Modern next-generation sequencing left physical models behind. The industry baseline systems infer a sequence optically, tracking synthesis byproducts by flooding a flow cell with fluorescent dyes and photographing the flashes of light as new nucleotides attach. Translating billions of distinct optical synthesis events into genomic data requires heavy hardware, massive camera arrays, and immense computational processing power. Oxford Nanopore Technology, however, completely abandons optical synthesis. Instead of building a complementary strand piece by piece to take pictures of it, this system takes a pre-existing single strand of DNA and pulls it through a microscopic hole, directly measuring the physical properties of the molecule. Exploring this system requires understanding how tracking sub-nanometer physical disruptions produces continuous reads over 100,000 bases long, and why interpreting those disruptions demands deep learning. Stripping away chemical synthesis and optical cameras removes the theoretical limits on read length, offering an entirely different method of genomic data collection. The hardware foundation begins with an engineered porin protein embedded within a 10-nanometer thick synthetic lipid bilayer. Its internal diameter is exactly 1 to 2 nanometers, precision-engineered to accommodate a single strand of DNA. This membrane separates two chambers filled with potassium chloride solution. When a 120 millivolt electrical potential is applied, it drives the negatively charged chloride ions strictly downward through the open pore. This unimpeded ion flow establishes a resting state, a steady baseline electrical current registering between 1 and 5 picoamperes on the live data graph. To put that in perspective, a single picoampere is 1/trillionth of an amp. The current powering a standard household light bulb is hundreds of billions of times stronger than the signal flowing through this channel. This baseline current acts as a highly sensitive microscopic tripwire, primed to detect any physical intrusion at the atomic level. Left to the physics of the voltage gradient, a DNA molecule whips through the pore far too quickly for any sensor to accurately sample. To solve this, a helicase motor protein docks precisely at the pore's upper mouth. As double-stranded DNA enters, the helicase unzips it, discarding one half. It acts as a brake on the remaining single strand, ratcheting it into the pore at a highly controlled, readable rate of 1,000 bases per second. Once the single-stranded DNA occupies the confined space inside the pore, its physical bulk obstructs the downward flow of the chloride ions. Because the specific bases, adenine, cytosine, guanine, and thymine, have different shapes and charge profiles, each one reduces the ion flow by a characteristic amount. You can see this in the live data output. As the bases pass through, the baseline current plummets, generating a jagged, fluctuating line between 0.5 and 2 picoamperes. In a physical sequencing environment, mechanically regulating the kinetic motion of the molecule is a strict requirement for electrical detection to work. Despite the biophysical precision of the setup, the raw, single-pass accuracy of these reads historically sits at a low 85%. This chart highlights the physical limit of the detector. When measuring individual bases, adenine drops the current to 0.8 picoamperes, thymine drops it to 0.9, cytosine and guanine sit tightly at 1.1 and 1.2. These target values are tightly packed. Once you introduce electrical noise in the detector, minor variations in pore structure, and slight mechanical slips from the helicase motor, these rigid bars blur into wide, overlapping bell curves. Distinguishing single bases with perfect confidence based solely on a fractional picoampere drop is mathematically impossible with this pore geometry. Furthermore, if the DNA strand slips through the helicase motor a fraction of a millisecond too quickly, the resulting signal is notably weaker, generating missed reads and compounding the final error rate. Pushing past the 85% accuracy ceiling requires reframing how the signal is interpreted, moving beyond single-base detection entirely. The physical reality of the nanopore is that it doesn't isolate one base at a time. The channel measures the aggregate obstruction of 5 to 6 bases occupying the space simultaneously. To solve for the ambiguity, the system uses a k-mer model. Instead of looking for 4 single bases, the target shifts to the 1,024 unique current profiles emitted by every possible combination of 5 consecutive bases. Deciphering overlapping continuous multi-base patterns cannot be done efficiently with standard linear algorithms. The raw data routes to deep neural network base callers. These models execute context-dependent calling, mapping the live signal against the k-mer database and using surrounding sequence context to mathematically lock in the identity of the central base. Applying this algorithmic context drastically narrows the margin of error, pushing the base-called accuracy from the raw 85% up to 98 or 99%. Achieving high-fidelity nanopore sequencing is an equal split, half biophysics hardware, half advanced signal processing. That remaining error margin consists of homopolymer indels, insertions or deletions that occur when the helicase motor occasionally stutters or re-grabs the DNA strand. In standard 1D sequencing, once the template strand drops through the pore, it is gone forever. The read cannot be verified. To correct this, scientists manipulate the library preparation, creating 2D duplex sequencing. A physical hairpin adapter is ligated to one end of the double-stranded DNA, permanently linking the template strand to its exact complement. During sequencing, the template strand is threaded through the pore to generate read 1. The motor stops exactly when it reaches the hairpin loop. The helicase then unwinds the hairpin adapter, forcing the loop flat. This allows the complementary strand to follow directly into the exact same pore, generating read 2. A consensus voting algorithm compares the two reads. A true biological mutation will appear in both strands. A random mechanical sequencing error will only appear in one, allowing the software to flag and discard it. Forcing systematic errors to cancel out yields a final consensus accuracy above 99%. Manipulating the physical topology of the DNA molecule before sequencing provides a reliable defense against the unpredictable kinetic movements of a molecular motor. Generating whole genome data requires parallel scale. A single Minion device runs thousands of pores, while higher throughput Prometheon flow cells operate nearly 48,000 nanopores simultaneously. Because this system pulls intact strands through a sensor, rather than chemically amplifying them, there is no PCR length bias to artificially truncate the data. Standard reads routinely reach 10 to 100 kilobases, and specialized preparations have yielded continuous, unbroken reads over 2 million bases long. Eliminating expensive fluorescent dyes and utilizing reusable flow cells significantly cuts overhead, driving run costs on portable devices down to $50 to $100. Furthermore, because there is no batch chemical processing, the sequence data is generated in real time, appearing on screen the exact moment the strand slides through the pore. Live data tracking immediately enables adaptive sampling, allowing researchers to instruct the device to focus on specific targets on the fly, or halt a run early to act on preliminary results during an outbreak response. The immediate hardware goal for physical sequencing is to bypass the computational k-mer bottleneck entirely. Future architectures propose achieving sub-nanometer spatial resolution by surrounding the pore with multiple detectors, an array of stacked electrode rings that scan the DNA molecule from several angles simultaneously. Capturing a detailed 3D spatial map of the current could allow the system to perfectly isolate and distinguish single bases, removing the need for 5-mer probability models. The continuous evolution of nanopore technology ultimately demonstrates that capturing the genome does not require synthesizing better chemistry. It relies on engineering precise physical sensors to read the molecular blueprint exactly as it exists.